Hi there. So I wanted to go over the lab handout real quick by means of giving you feedback on your answers that you gave. So I've graded the questions in the lab handout, but instead of giving you a filled out lab handout, I'm just going to go through the answers and what the right answers were going to be. So let's just scroll down to where the questions were. So you remember we did the 4 different tests, right? The very first one was the Benedict's test. And the Benedict's test tests for reducing sugar. You'll notice there's always more than one tube here, and that's because we need positive and negative controls. So the first tube is always just the regular, the solution itself with just distilled water. And so we would expect that to always be negative because it doesn't have anything in it. In this particular case, and in most of the cases, you have something that has glucose in it, which is, of course, a monosaccharide, something that has starch in it, which is a polysaccharide, a complex carbohydrate, ovalbumin, that's your protein, and corn oil, that's a lipid. So these compounds are actually your positive controls, because if Benedict's comes back positive with glucose but not starch, that's positive for reducing sugar, because remember, glucose was a reducing sugar, but starch is not. And ovalbumin, which is a protein that doesn't have reducing sugar in it, should be negative, and corn oil is also negative. You see, some of you noticed that there's 2 layers in the corn oil. That's because oil and water don't mix. So most of the solutions we have are watery solutions. And because most of them are watery solutions, they don't mix with oil. So which of these tubes is the negative control? So the true negative control would actually be 1, right? Because it's distilled water, because we know what the outcome is going to be. But all 4 of these are negative, right? 1, 3, 4, and 5. So the negative color is obviously blue. The tube that has evidence of reducing sugar is 2, and it looks orange. The tubes that don't react with Benedict's reagents are 1, 3, 4, and 5. And the evidence that this test is specific for reducing sugar is because you have the starch and the glucose next to each other. You have all these different macromolecules in the different tubes, but it only shows up for reducing sugar, and it doesn't show up for any of the others. So that tells you that it's specific for reducing sugar. The next one, Benedict's, we looked at the ability to discriminate between a high and a low amount of sugar. So you've got the 10% glucose, which obviously is higher than the 1% glucose, and then you have the sucrose. Sucrose is negative because it's not a reducing sugar. The difference between tubes 2 and 3 is a color difference, obviously, but it's related to the concentration difference. Remember that Benedict's turns is kind of like the penny, kind of backwards, right? So the penny goes from red to green to blue, and your reaction is backwards. You're starting at blue. With a little bit of reaction, you get to green, and with a lot of reaction, you get to orange or red. Sucrose is a sugar, but sucrose is not a reducing sugar, right? All right, then the test for the polymer for starch. So for iodine, again, you've got the distilled water, you got the glucose, you got the albumin, which is your protein, and you've got the corn oil. And note there's 2 layers again. And the only thing that's positive here is black, which is the starch. So iodine is an indicator reagent for a sugar polymer. So evidence for the sugar polymer is 3, and it looks black. The tubes that don't react are 1, 2, 4, and 5. And the test that the evidence that the test is specific is because it only reacts with starch. It doesn't react with glucose, which would be another sugar. And the reason why there are 2 layers is because it's a lipid, and lipid and waters don't mix. Now the protein is burette. Burette is in the ovalbumin, so the ovalbumin, that's your protein. which tube is evidence of a peptide bond? Number 4. What it looks like? Purple. And what is the evidence that this is specific for a protein? That's because it didn't show up as purple as in any of the other macromolecules. So it doesn't show up as purple in the case of glucose, doesn't show up as purple in the case of starch, and doesn't show up as in the case of corn oil. And you can see 2 layers here if you look really closely. So because again, it's a watery solution. The next test is the test for lipids. Which tube is evidence for a lipid? It's the last one, the corn oil. And in this case, our Sudan 4, and I mentioned that in the lab handout somewhere, our Sudan 4 is dissolved in an oil. So oil mixes with oil, and so that's why there's not 2 layers here, but there's 2 layers in all of the other ones. This red layer right here in all of these other ones is actually the same. It's literally your reagent that doesn't mix with anything. Now, when you're doing this in real life, you actually have to flick the tube to make sure that it mixes. And there's actually a typo in here. What is evidence that this test is specific for protein? It's not specific for protein, it's specific for lipids. And it shows you that only the one, number 5, gives you a positive reaction. And then we get to the unknowns. The unknowns, so you're just literally looking through here and putting positives and negatives on here, right? So for this unknown here, you've got a positive on the Benedicts, negative, negative, negative. For this one here, you've got a positive on the Benedicts, negative, positive. And this one looks like it's positive because it looks like it's sort of mixed in here. This one here is negative, negative, positive, negative because you've got 2 layers. This one is positive, negative, 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 positive, negative, 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 positive, negative, 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 positive, negative, 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 positive, negative, positive, negative, 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 positive. And then I gave you choices based on what your results were, where you had a couple of different choices where you had to pick between the different options. Limitations of some of the tests, like for example, for the Sudan 4, you've got to mix it. If you just slowly layer it in there, it's not going to show up as a mixed layer. So some of these things you've got to be a little careful. But that's basically a quick run through your lab.